What I thought I'd do today is go through this paper so that we can see how to improve our knowledge. So if we are looking at the questions here, we're going to start with the first question, and it is asking about, do you know which of these statements describes a problem that's resulted from the increase in antibiotics? Now, whenever I do any of these questions, the first thing I want to do is I want to identify some of the concerns that I have with the knowledge being brought forward to the question. So first of all, it's asking about antibiotics. So then I would say to myself, right, well, what do I know about antibiotics? So I should know that, hi, Chidima, how are you? I'm going to actually do some questions right now. If I'm doing antibiotics, I am going to be therefore noting to myself that we are looking at the bacteria will be killed because of the antibiotics. I'm good, thank you. I am good. And then what we need to do is we then need to look at what have they actually told us in the question, because they want to know which statement is going to describe a problem. So this is important that we're noting that it's a problem with the increased use of antibiotics. So you should know antibiotics are going to kill bacteria. We should know, therefore, that bacteria that become resistant to the antibiotic we call resistant strains. So we can just add in a little note to ourselves. Resistant strains are not affected. So I'm just writing some notes to myself. I'm not answering the question yet. Yes, I am on my YouTube channel right now. I thought I would show the 2023 AS paper and work our way through that one if you want to pop over and see it. So yeah, so what we've got here is me just writing some notes to myself. So when I'm doing practice papers, when I'm going through, I would be giving myself little hints as to how I could answer the question. I'm pulling that information forward so that I can help myself. So then I will then go back to the question and actually say, right, well, what's it asking me then? So penicillin can no longer be used to treat infections. Well, we know that's not true because they are going to be used to treat bacterial infections. So as long as they are going to kill off the bacteria, whichever they are effective against, then that is a positive. It's not a problem that would result from the increased use of antibiotics because the penicillin will still be killing the bacteria. It just depends how much it's going to kill them based on has their DNA been mutated or not. So let's look at the next one. The rate of MRSA infection have increased. Well, do you understand what MRSA actually means? Do you understand that that is methicillin resistant strain of Staphylococcus aureus? So Staphylococcus aureus is the type of bacteria, and that bacteria, the genus and species Staphylococcus aureus, that is going to be killed if it is not resistant. So if we're talking about the rates of MRSA, we're talking about methicillin resistant strains of Staphylococcus aureus increasing. That would be a problem that has resulted from the increased use of antibiotics. So I'm thinking that this is the right answer, but I'm going to double check that it's the right answer by going through the other wrong answers. Because if we're doing the multiple choice section, we should be able to identify what's wrong so that we're only left with that right answer. So some bacteria and fungi have evolved resistance. Well, we know that fungi do not evolve resistance to antibiotics because fungi are not affected because it kills prokaryotic organisms, and bacteria, I'm going to note this up here, are prokaryotes. So we know it's definitely not C. And then if we look at D, then what we can see is viral infections can no longer be treated with antibiotics. Well, viral infections were never treated with antibiotics because, again, going back, it is about bacterial colonies and it's about prokaryotes. So let's just double check. Penicillin can no longer be used to treat infections. That is incorrect because it's no longer able to treat infections caused by a resistant strain. So therefore, leaving us with the only right answer, which is B. Okay, let's move on to question 2 then. So the diagram shows a computer model of the structure of an antibody. Now, you should know when you're doing your module 4 content that your antibody is made up of 4 polypeptide chains. And those 4 polypeptide chains are held together by disulfide bridges, and we have a quaternary structure. So straight away, when I start to look at this, I am trying to give my brain a little bit of insight by doing a quick recap and blurting out the information that I know. So I already know that I have my antibody. I know that that antibody is a protein which has a quaternary structure. And I know it's made up of 4 polypeptides. And I know those 4 polypeptides are made up of 2 light chains and 2 heavy chains. Now, I know I'm not answering this question yet, but what I'm trying to do is I'm trying to just help myself do that revision of a topic area. So when you are doing revision, I would advise you to not just look at the question when it's multiple choice and think, right, how can I answer? Is it A, B, C, or D? Oh, I know it's definitely this one, and go from there. What I would advise you to do is to actually use this time that you have to see if you fully understand the topic area, to see if you have all that understanding, that if they ask any question, you can then go from there. So you should know that you have got your constant region. So we're going to use the information they've told us. So the constant region that we have in our amino acid chain is actually made up here. So this is my constant region, and that is constant because that therefore has the sequence of amino acids which are always remaining the same. And remember, if we're talking about proteins, we're talking about polypeptides. These polypeptides therefore have a primary structure, and that primary structure is the sequence of amino acids. And that sequence of the amino acids is the same. So again, I'm not answering the question yet. I'm just priming my brain so that I know what it is that they are actually assessing me on. So if we're looking then at this question, it's asking about my antigen binding site. So I know that there is I'm going to add it down here, a variable region on my antibody, and that variable region is variable because it has a different sequence of amino acids. And the fact that it is a antibody and that is an immunoglobulin tells me that it has a globular structure, and that globular structure gives it a specific shape. And that specific shape, that specificity, means that this antigen binding site will have a complementary shape. Let's get up here. Complementary shape for binding with that antigen. And so what you are looking at is whereabouts is this going to be? Well, I can tell you straight away, the variable region is not going to be down where the constant region is. So we can eliminate D straight away because we know that variable region where we're going to find our antigen binding site isn't within the constant region. So that's the first way to eliminate. Now, the next thing we need to think about is, well, where is this site that they're going to be able to bind with the antigen? And the antigen can be either on the surface of the pathogen, or it could be a toxin that they've produced. So what you have to note then is that this is actually, let me change color. This region up here would be my antigen binding site and therefore my variable region. And because of that, what we know is that that would result in A being the correct area where you would find the antigen binding site. And that's because B and C are too far into the molecule, so therefore they wouldn't be the place that the antigen would be able to bind to. Okay, let's move on. So let's just write our answer in there, which is A. Okay, so now we're looking at Simpson's index. Now, Simpson's index of diversity is very specific depending on the exam board that you are sitting. So I'm looking at OCR A here, so they have a very specific Simpson index of diversity equation, which you will be provided with in the exam. So if you are provided with this in the exam, they are expecting you to not only be able to use it, but they're also expecting you to understand what it means. So they've told us that it can be used to estimate the biodiversity of a habitat. The scientists calculated D for a grassland habitat. So we've been told we've got a grassland habitat. 1 year later, the scientists then calculated that again for the same habitat. The number of plants recorded for one of the species increased from 100 to 400. All the other values remain the same. So because we have all the other values remaining the same, that therefore tells us that we have the same species richness because the numbers haven't changed. So we know we've got the same species richness because they've told us, although the number of one of the species has increased, so you can see that there's been an increase there, that was for one of them, all the others remain the same. That tells us there's the same number of species, so that would mean that it's not A. So we can eliminate A straight away because we are being told that the species richness isn't increasing because the number of plants for one of the species increased, all the other values remain the same. None decreased, they remain the same, so we have the same number of species, so our species richness has stayed the same. So now we need to think about what's going to happen to our diversity. So if we are increasing the number of individuals, that could affect the evenness. So we've increased, and we haven't increased the evenness, we've affected the increase, we've increased the number there, but we could have affected the evenness. Now we don't know the numbers, so therefore we need to consider what that might mean. So we don't know the numbers of the other individuals, and because we don't know the number of the other individuals, we can't say whether or not that would definitely increase or decrease. So it's a really interesting one here, because actually it depends. I think there could be two answers that are correct for this. So if we're looking at the number increasing, do you know what the little n stands for? Do you know what the capital N stands for in your index? So capital N stands for the total number, and the little n stands for the individual for each of the species. So therefore, if you understand that, we can eliminate another answer here because we know that the value of n is not going to decrease, it's going to increase because we've added 300 individuals. So because we've added 300 individuals, that is therefore going to increase that value rather than decrease it. Now, because we don't know the number, when it comes down to the other species and how evenly spread they are, I would say that it could technically be either B or C, because if it is affecting the evenness, then that would potentially decrease the value of D. However, if it's not, then the biodiversity would potentially increase. So actually it could be an argument for and against on this one. And therefore what you'll find is when you look at some of the mark schemes, you will find that the answer is they want it to be this, and then they said, or it could be this. And that's because when the examiners have come together and they've looked at the question, they've realized it could be more than one which is correct, depending on how you look at it if they haven't given you enough information. So it's always important to understand that there's not always one answer that is correct. There can be other answers as well. So do make sure when you are doing this revision, when you're going through these topic areas, that you are looking at not only the mark schemes, but also understanding why there could be two answers that are correct. In this case, B and C are both correct because they haven't given us enough information. We don't know the values of the other species to know whether that's affected the evenness. Okay, let's look at question 4 then. What is an example of in situ conservation? So this is important that you note the different types. There's in situ, and the other type that you should know is ex situ. So if you know there's two types, you've got in situ and you've got ex situ, then this one is asking specifically, do you know what's the in situ one? So the in situ is inside the natural habitat. You're conserving the habitat, you're conserving the individuals where they are found. Ex situ, you are removing them from their situation, you're removing them from where they're naturally found, and you are going to do all you can to conserve them, whether that be in a botanical garden, whether that be in a zoo, or whether that be in a seed bank, if we're looking at plants and we're looking at how we can maintain biodiversity by having the seed bank. So therefore, straight away, I can remove A, I can remove B, and I can remove D, because these are all examples of ex situ conservation, and therefore that leaves me with the only right answer, which is a marine conservation zone, a zone within an area, a marine area, so where we could have a area set up in the ocean, for example, where we are conserving that place to make sure that those organisms are being maintained where they are. So it's in situ conservation because it's in their natural habitat. We've not removed them, therefore in situ. Okay, so let's look at this one then. It's really interesting. This paper, so this is 2023, paper 1. This one is showing you very much where they're assessing the module 4 content first through these multiple choice questions, which means that later on, they're probably going to look at other areas. And in other years, they will switch that around. Okay, so the giraffe is a species of mammal with the species name. I'm not even going to try and say these ones out loud. So we have been told that this is my species name and this is my genus name. So I know the generic name, the genus goes first and the species goes second. The next thing I also know, hi Ghost, yeah, I know, results day in a month. Is it exactly a month? It's crazy how quick it'll come around. It's the 15th, isn't it? So what have we got? 1, 2, 3, 4, technically a bit longer. So 14th, sorry. So you've got, yeah, just longer than a month. Or, yeah, 5 weeks. 5 weeks tomorrow. Which is crazy. Okay, back to this question. What's the correct format for the binomial naming? So therefore they are checking, do I know how to do my naming? So I know the genus and species always go in that order. And then the next thing I need to understand is that the genus should always be written with a capital. So as they've shown it here, it's a capital G, and the species should always be with a lowercase. So therefore now I need to look at, well, they've got these the wrong way around. A and B are the wrong way around. Therefore I can eliminate those two. Now I need to look at, well, whether or not it is C or D. Now, if we are looking at abbreviating, then yes, we can technically use an abbreviation of just doing G dot and then having the species name. However, because it is actually typed format, it should be in italics. Now, there's no italics in C, so therefore that would say that C is incorrect, and they've used the full names for the naming, which should be what we use. When we do the abbreviated form, it's normally because we are trying to make it quicker. So it's important that we do note that when we do this, that just because in this exam, they have done it as being the full giraffe and then the species name, they could in another question do it the opposite way around, where they give it to you with a capital letter dot and then the species name. That is still correct as long as it's in italics when it's in the format of typed. So for example, we talk about E. coli, which is where we have abbreviated that down. So it is a thing that we do. And when we talk about Staphylococcus aureus, we tend to do aureus. So it is important that we know that that is how we can do the naming, but make sure that you double check because it's asking about the correct format. So it wants to know, do you know it should be in italics as well as genus, capital letter, species, lowercase. Okay, so looking at this next one then, which kingdom species or which kingdom contains species that are heterotrophic and composed of hyphae? Oh, I'm glad I'm giving you a bit of nostalgia. I thought I should actually get back and do some real study with students again, because obviously, although you guys who have finished cohort 2025 all finished, those who are doing it 2026, you still need to keep that going. And the worst thing you can do is switch off and not look at things again. The number of times that my students will go off in their summer holidays and they will be like, oh, it's fine, I've got lots of time, summer holidays, and they'll come back at the start of September and have forgotten the content that they learned beforehand. So if you're cohort 2025, great, have that break, have that downtime, enjoy yourself. Oh, thank you. I definitely want you guys to do that, but I have to kind of move on to the current cohort 2026 because actually I still had so many of you come to me and say, how have I only found you just before my exam? And I don't want that. I mean, my channel's been going now for over a year, and I want to make sure, and I need to make sure that enough students know that these resources are there because it's being videoed, because it's on my YouTube channel. You can go back and watch it, whether that's September, whether that's December, whether that's just before the exam. This will be here to guide students, and I'm only using the papers which are obviously open for you to have access to, because you can go to OCR A's website and actually download them yourself, because actually that's the right thing. All the others are in lockdown. They should be in lockdown. Although I've seen them, I don't want to disadvantage anybody, whether that's to do with your predicted grades for UCAS or whether that is even just to know where you're at right now. These mocks that you do and these practice papers are great for giving indications of where your current knowledge is and also identifying where you're lacking knowledge. But the one thing I would say is too many students memorize mark schemes for these topic areas. And that's not what I'm going to do. I'm not going to just tell you what's the right answer because that won't help you. It's about how you are able to use the knowledge that you have and apply it to the question so that if they give you it in a different order, you know how to answer the question. I know, well, this is the thing that When I started teaching OCRA a couple of years ago again, so I've been teaching IB for many years because I've been the head of department and I worked my way up and whatnot. So I'd done a lot of IB, and then a couple of years ago when I decided to change my career path so I could spend more time with my daughters, I went back to teaching A level again and teaching OCRA, which I hadn't taught for many years, and I couldn't find resources for myself. So this was the thing that I found really interesting, is that there weren't resources for teachers coming in to teach the course if they hadn't done it. So anyone who has gone through university, doing a PGCE and is about to become a teacher for biology, for example, it is nerve-wracking when you're standing in front of a class, and it depends how big your school is. I had a class of 35 students in my A-level class because that's how big the cohort was, and I had to teach a lot of students in one class. Now, if you're very lucky, I also taught in a school where there was 9 students in the class, and that's amazing. So I have had the vast array of teaching big classes, small classes, but then also as a teacher, you've got other year groups, other topic areas, other things to do. And if you don't have the resources there, it can become quite difficult. So I found that there wasn't any OCRA stuff out there at the time when I started. And it was a couple of my students who said, Kristen, you should put some stuff out there. And so I did. And this is where this all came from. Students telling me what to do. And I went, okay, I'll give it a shot. How would I recommend planning biology revision? I would say the best thing you could do over the summer is not bombard yourself with lots of things to do, but just slowly drip in a quick test here and there. And when I say that as a quick test, what I mean is doing like 10-15 minutes, just double check you remember biological molecules, make sure you remember nucleic acid nucleotide, just do a little bit of assessment. Oh, what was that topic area again? Oh, I just quickly look through my flashcards, or I'll quickly look at a mind map I've made, or I'll do a AIQ chat question, set of 10 questions on it, whatever that may be. What I would suggest you do is have the small drips of it over the summer. I'll watch one of Christine's videos on YouTube, for example. Whatever it is that you do that you find useful for you, I would say doing that, but absolutely nail module 2. So if I were to say to any of my students, what should you do over the summer? Make sure you're coming back in September absolutely solid on module 2, because module 2 basically just gets put into place for all the other topic areas. So whatever you are doing, you will see where module 2 comes through. So if you're doing respiration, for example, it's all about biological molecules and biological membranes and things moving and enzymes. And if you're looking at photosynthesis, again, you're looking at biological membranes, you're looking at biological molecules, you're looking at movement of substances. So it's important you understand those fundamentals, those building blocks, so you can then move on and get the rest of it. What's my predictions for grade rounds? I think they're going to be absolutely the same as they were last year, if I'm honest. I've been going through them again, actually. I was doing that today before I saw one of my clients this morning. And when I was looking at the difference between, hold on one second, let me actually just do some quick calculations, because I didn't actually do them for the last 4 years. I did paper ones. So 2024 or So when you look at the actual grade boundaries, to get A star in 2024, you needed to get 69%. To get an A star in 2023 was 68%. To get an A star in 2022 was 68%. And to get an A star in 2021 was 63%. Now you've got to remember 2021 was straight after COVID. So things were very different then. And so because of that, hi Ella, because of that, what I would say to you is the grade boundaries have been pretty stable for the last 3 years. So I feel like they're going to be around the same areas. To be honest, where it differed the most was down towards the end. So an E grade in 2021 and 2022, you only had to get 19 out of 270. No, you only had to get 52 out of 270. So 19% to get an E grade. Whereas last year it was the highest it's ever been. It was 24%. So it was those bottom end students who were actually at more of a disadvantage than the top end. The top end hasn't really changed that much. So it's always been like an A, 59, 57, 58%. It's about on par. 50, 48, 48. They're pretty similar grade boundaries over the last 3 years. So I feel like having looked at the papers and seen how they compare with the other exam papers, I don't feel like they're any different. So I feel like the grade boundaries will probably be about the same. There'll maybe be 1 or 2 marks where it'll move slightly. But apart from that, I don't see it changing that much. But I could be wrong. Who knows? I'm not marking this year, so I'm not part of that process. So I'm not entirely sure. It really depends on the cohort who sit it and how accessible those papers were. But I would be surprised if they changed dramatically. Right, let's carry on with this question. This question actually was really interesting because it started off automatically. It already told me that it had hyphae. And so because it already told me it had hyphae, I knew straight away that it was a fungi. So the kingdom, you obviously have your 5 kingdoms. You've got your prokaryote, animalia, you've got fungi, protoctista, you have plantae. So we know that they are composed of hyphae, so they can't be plants, they can't be prokaryotes, and they're not protoctistas, therefore leaving us with the only right answer, which is fungi. Okay, let's move on to this next question then. So a student observed a xylem tissue under a microscope. Now they love checking your practical skills. So this is where they're actually assessing you on not only module 1, but also module 2. So this is where what you have to do is make sure when you are revising that you're using that specification, you're referring back to that specification so that you know where this is actually being assessed and what it's assessing. So some students feel like they don't get a lot of assessment in one topic area, for example, and they're like, oh, this exam paper barely touched on that topic area. And what you'll find is that the topic areas are sometimes interwoven. And so that's what you need to know as you're going through and doing that revision, that you have looked at the specification alongside. You don't understand question 8. Ryan, give me a second. Let me do question 7 first, and then I'll go to question 8 and explain it. So this one is telling you that you have got a eyepiece graticule, and I can see this is my eyepiece graticule up here. I'm then told that I have got a stage micrometer scale down here, and every division is 10 micrometers. And we're told that the xylem vessel has a diameter of 21 units on my eyepiece graticule. So I'm going to come up to the top and I'm going to look at where 21 is, and I'm going to bring this down here so I can see quite clearly that's in between 6 and 7. This is a really nice, easy question if they're going to ask this type of question. And all I basically have to do is do 7 times 10 to tell me that the actual diameter is 70 micrometers. This is really lovely, a question, if they ask it to you like this. Right, question 8 then. So Ryan, you said you don't understand question 8. So you've got a pathway And it's saying that the pathway is taken by a protein. So they've given you hints in the question. So a pathway taken by a protein that has been made and is secreted from a cell. So there's some hints in here. So first of all, they've told me I'm dealing with a protein. Well, the protein I know is made at the ribosomes. So then I need to think, right, well, what type of ribosomes do I have? Well, I've got free-floating ones. So the free-floating. So they are just found in the cytoplasm. And I also have those which are attached to my ER. So therefore, I've got my rough endoplasmic reticulum. So because I've got my ribosomes, which can either be free-floating or part of my rough endoplasmic reticulum, they're telling me that these are going to be secreted from the cell. So therefore, they are going to need to go into a secretory vesicle. So because we're going into secretory vesicle, again, little hints in the question. And that's what you need to be able to do, is note where those hints are. That's the thing about these exam papers, is they do actually help you if you know where to look. So Ryan, I'm glad you find that this is making sense now. So therefore, could you tell us what is the right answer? So straight away, secretory vesicles, we should hopefully know that that means we're using our rough endoplasmic reticulum. So because it's that, we can eliminate A and B. And now we're down to a 50-50 chance. So now it's whether or not you know the way in which they go together. So what's the difference? We've got our rough endoplasmic reticulum with vesicles going to our Golgi apparatus, to vesicles to the plasma membrane, or rough endoplasmic reticulum to the vesicles, the smooth endoplasmic reticulum to vesicle. Well, we can say straight away it's not D and that it's C, because we should know that the smooth endoplasmic reticulum is for the manufacturing of lipids and carbohydrates. Whereas this is only mentioning the fact that it's to do with the protein. Now, if the smooth endoplasmic reticulum is going to be making, let's say, the carbohydrate that's going to be attached to the protein, so if I had a glycoprotein, that glycoprotein would be made in the Golgi apparatus. So this is what we are talking about when we say that the proteins can be modified before they're packaged. So that modification would be the adding of the carbohydrate to the protein to give me a glycoprotein. So that would happen at the Golgi apparatus. You don't understand any of this? Well, I'm hoping that the more you go and watch my videos, the easier it will become. A-level biology is tough. And because A-level biology is tough, it's about going over it again, 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 until you feel confident with it. Lipid or carbohydrate or both, it can make both. So it just depends on what it is that the smooth endoplasmic reticulum is needing to produce, whether it's making lipids, whether it's making carbohydrates, it really just depends. Okay, let's have a look at question 9 then. Okay, so question 9, we have got a skeleton. I love the cytoskeleton. This comes up more and more, the cytoskeleton. They like to assess you. So what's the role of the cytoskeleton? Now, straight away, when you start talking about cytoskeleton, you should start to think about all the things that it can do. It can maintain the cell shape. It can allow the cell to move. It can move the organelles within the cell. It can be used as making the spindle fiber so that they can attach and pull the chromosomes apart. Now, what's really interesting with this question is they've specifically said human skin cell. So straight away, they have given you a very specific type of cell. There's a reason for that. That means that there's something that you need to identify in these answers that is relevant to that skin cell. So, Chidama, when it comes to hormones, hormones are involved in lots of different things, but basically they're signaling molecules, and what they signal depends on what happens with the binding with the receptor. So it could be that the signaling molecule might switch on genes, and those genes result in the production of proteins, those proteins being potentially enzymes, those enzymes needed to build the lipids or the carbohydrates, or what depends on what it is that the cell needs to do. So when we talk about hormones and we talk about whether they are going to be used, it depends whether or not we're talking about a non-polar, so a steroid hormone, for example, or whether we are talking about it being a peptide hormone. So there's lots of different types of hormones. So it's about, again, looking at the question that they are asking as to what answer you want to give. So yeah. Right, where were we? This question here then. So if we're looking at this, what have they told us? Right, movement of the cell through its environment and providing mechanical strength to the cell. Right, straight away, I know the fact that it's telling me it's a human skin cell, movement of the cell, the skin cell doesn't move. So that can't be the right answer. So A is wrong because we're talking about a skin cell. Movement of the cell through its environment and transport of organelles within the environment. Again, we're talking about a skin cell that's not going to be moving, so that can eliminate that. So if they had changed this and they'd made this, for example, a lymphocyte, or they'd made this a phagocyte, probably phagocyte's more relevant to this for the movement. So if they said, for example, a phagocyte, then that movement, the cytoskeleton being used for that movement of the cell through its environment would make sense. But they haven't asked about that. They've asked about human skin cell. So then let's look at the next one. Provide a mechanical strength to the cell and transport of organelles. Well, that does make sense because we know that it's going to transport the organelles, and we know that it's a skin cell. So we need that mechanical strength because that skin cell is acting as a barrier to the external environment. So we want that barrier to be there. And then let's have a look. Supporting the cell wall. Well, straight away, cell wall, that's only found in plants or fungi or prokaryotes. It's a human cell. They've told us it can't be D, leaving us with the only right answer, which is C. So as you're going through these, as I keep saying to you, make sure you're taking the time to look at what is actually being asked and what hints they've given in the question as well. Okay, so which feature is associated with a prokaryotic cell? Cell walls which are made of chitin? No, they are not. We know that they are made of peptidoglycan. And for those of you who are just joining me, you'll notice that your teachers will say things slightly different to the way I say things. When they're naming things, they'll give them the correct name, whereas I have to be very specific because I struggle with my spelling. I struggle with how the letters come together, that I will say things how I sound them out because that helps me to be able to write them in the right way. So I may say something one way and your teacher pronounces it another way. As long as you are writing the correct answer down with the spelling, that is what's important. I always say, and I make a joke, well, I'm Scottish, so I can say things whichever way I want. I had to train myself how to say a Bunsen burner when I first came to England to teach, because I used to say Bunsen burner. We're going to light the Bunsen burner, and all the kids would be like, Miss, what's a Bunsen burner? And I was like, oh, sorry, it's a Bunsen burner. And I had to train myself to not be as Scottish as I was when I first came down. So yeah. Little sidetracked. For those of you as well who don't know, I do go off on little tangents when it comes to me doing these free lives, because that's the point. This is just a fun thing that I do for free to help. But if you are someone who is coming to my group tuition sessions, then they are not quite as trying to think of what the right word is, fluid and going off in different directions as they are during these sessions. But anyway, back to this one. Right. Associated with prokaryotic cells, remember we know prokaryotic cells are bacterial cells. So extra chromosomal DNA that's circular, well, that's basically describing a plasmid. So that one I would say is correct. Linear DNA that forms, well, we know it's not linear because linear is found in eukaryotic organisms. So we can remove those two. And now we need to know with the last one, 70S ribosomes are found in prokaryotic cells, but endoplasmic reticulum is not, therefore eliminating D, leaving us with the only correct answer, which is B. So again, it's about eliminating those wrong answers that leaves you with the only right answer. And that's how you know your content. The more you practice that, the more you actually understand how useful multiple choice questions are. And that's why a lot of the universities, if you're going to the likes of Cambridge or if you're going to the likes of University in America, they will use multiple choice questions because it's about them being so close to almost being right that you have to really know your content to be able to eliminate the wrong answers. And that's why the more you practice it, the more you do that, the stronger you will get, not only at increasing your knowledge, but also at figuring out what you don't know. And that's the best thing you can do. Figure out what you don't know, because that will allow you to then go and do the work to fill those gaps. And by filling those gaps, when you get asked questions about it, you will then be able to excel at those questions. Whereas if you're just doing the content again, and just going over the same stuff all the time, that's not going to help you to know what you don't know and to improve on what you don't know. And you also have to think about the course holistically, which is hard right now because in year 12 it doesn't feel as connected as it should do when you get to year 13. And year 13 is about to explode with the amount of content you're about to learn. If you haven't already started year 13 content, then it is going to be at such a fast pace that you have to keep up with that content. So I would definitely say, do make sure you are going over the year 12 content over the summer holidays so that you feel a bit more confident. But if you haven't, it's okay. There's still plenty of time. That's the one thing I would say to you. There is still plenty of time. You've got a year left, short of a year, before you sit your real exam if you are currently the cohort of 2026. But know that you're going to have so many things in that first term that are going to be taking your attention off to do other things. So you need to try and keep a revision timetable going, which is why I would suggest having some form of revision plan in place or from the start. Little and often is more successful than cramming at the end. Right. Anyway, let's see if we can finish these questions before I need to go. So which statement is a correct description of a polymer? So we're looking at polymers. Now, a polymer is broken down by, well, no, it's not broken down by a condensation reaction. It's broken down by a hydrolysis. That's quite a simple one to eliminate. A polymer is formed when two monomers are bonded together. No, that's a dimer. So that's not a polymer. Poly means many. So if we're talking about a disaccharide or a dipeptide or a monoglyceride, actually, no, I need to think that one. So you've got monoglyceride, which would be two things held together if you've got your glycerol and your fatty acid. So that would be a monoglyceride, yeah. Okay, so coming back to this one, so we're looking at all polymers are classified as either carbohydrates or proteins. Well, we know, just to be clear, I'm going to write this up here, that lipids, i.e. a triglyceride, is known as a macromolecule and not a polymer. So I know that that shouldn't be in my list. So that is true, it should be in my list, but it says all polymers. And the fact that it says all polymers is making me go, all of them? Well, what about DNA? What about RNA? They are polynucleotides. So therefore, they are many nucleotides held together. So they're polymers, just like a carbohydrate, whether we're talking about glycogen or starch, just like a polypeptide. These are polynucleotides, many units held together, many nucleotides held together. So therefore, I would say C is incorrect, which should leave me with the only right answer, which would be D. So some polymers are composed of several monomers that are similar in structure, but not identical. Well, yes, if we're talking about the proteins, proteins are a chain of amino acids, and we know that there are 20 variations. And so because we know there are 20 variants, we know that they're not going to be identical. So therefore, that would mean that D was the correct answer. Okay, a scientist analyzed and recorded all the chemical elements in one particular amino acid. So they are asking about which one that they are. So all the chemical elements in one particular amino acid. So what do we have? Right, so if we look at A, if we're thinking about an amino acid, we know we've got our carbon, which is our carboxylic acid group. We know we've got our hydrogen attached to the carbon, and we know we've got our amine group. So if I just quickly draw out the generic structure, so right now we can say straight away, looking there, it can't be A because there's no oxygen present in that list. So because there's no oxygen present in that list, it can't be A. So if we're looking at B then, so a carbon, hydrogen, oxygen, and nitrogen, well, we don't know what's in the R group yet. So we need to think about this and see if we can eliminate the other answers, because technically it could be that the R group is literally just one hydrogen atom. And if it is just one hydrogen atom, then that could be correct. But let's have a look and just double check. So if B is the right answer, that would therefore mean that we can eliminate C and D. Well, straight away, we can see carbon, hydrogen, oxygen, nitrogen. The only difference is that it's got a phosphorus there. Well, we know it doesn't, so we can eliminate that because we're talking about amino acid. And again, it's got phosphorus here, so we can eliminate out D, leaving us with the only right answer, which is B. Okay, looking at 13 then. With 13, we've got which description of the structure of a glucose molecule is correct. Okay, so glucose, I know, is C6H12O6. So straight away, I'm giving myself little hints. It contains 5 hydroxyl groups and has a carbon to oxygen ratio of 1:1. So carbon to oxygen ratio is 1:1. Yes, 6:6. So that is correct. It contains 5 hydroxyl groups. So let me just think about this. So I've got oxygen attached to my carbon, 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 attached to my carbon. So here I have carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. I have a hydroxyl group down here. I have a hydroxyl group down here. I have a hydroxyl group here. I have a hydroxyl group here. I have a hydroxyl group up here. 1, 2, 3, 4, 5. So therefore, I would say that at the moment, A is correct. So if A is correct, that must mean that I can eliminate B, C, and D. So it contains 6 hydroxyl groups. Well, no, it doesn't. Contains 6. Straight away, we know it's 5, so we can remove B and C because they both said 6. Oh, no, hold on. That was me jumping too quickly there, thinking it was saying the same thing. Definitely eliminate B, although this part, the carbon to hydrogen ratio is 1 to 2. That bit's correct. The first bit is incorrect because we've already counted that there is only 5 hydroxyl groups. It contains 6 oxygen atoms and has a carbon to hydrogen rate. Right, okay, so this is the bit that's incorrect, whereas this is the bit that's correct. Now, don't get caught out with what I just did there, and I skim read. Because I skim read, it meant that I didn't quite see the bit that was saying that it's 6 oxygen atoms. I read it as being 6 hydroxyl groups again, and sometimes that does happen to students in the exam. So just be aware that if you are the type of student like me, so I'm doing this live with you right now, and the reason I do this is to show you that even I've been teaching this for 20 years, I can still make mistakes because I skim read, I don't take the time, I think I know what it's asking me, and I start answering the question. So what I would advise you to do is to take your time going through these practice questions, going through these past papers. The more you can do, the more you can work out how to answer the question, the more you will find the next set of papers that you get given are just seem a bit easier because you've had that exposure. That's why you do mocks, but mocks will only get you so far. Try and do more than that. Okay, let's look at this last one. It contains 12 hydrogen atoms. Yes, that is true. And has a carbon to oxygen ratio of 1 to 2. No, it does not. So that part is wrong, which therefore leaves me with the right answer, which was A. Okay, moving on. Which description of the structure of cellulose is correct? Right, cellulose. Again, I'm just going to write little notes myself. It's a beta glucose. So it's beta glucose. I know that it's got 180-degree inversion. I know it's got cross links, which gives it its tensile strength. I know it's part of the cell. So one of the things I would say to you as well as you're doing this and you're doing revision is start making your little flashcard notes that you would say on any topic area to do with cellulose. So this is a multiple choice question, but if they flip this into a 2-3 marker, I've already got my go-to answers, and then I just need to relate it to whatever the question is that they are asking. Okay, so alternate alpha glucose monomers rotated right. They've tried to jump straight in and confuse me because I saw the 180-degree inversion. If I didn't take the time, I could get caught out in the fact that they said alpha glucose rather than beta glucose. It's a branch polymer forms from beta. Well, it's not branched. We know that it's not branched because it's cross links between the fibers. So therefore we know it's not B. So if we're looking at this, then we're down to a 50-50 shot. Hydrogen bonds form between the coiled polymer chains. Well, it's not coiled. So therefore, it's not coiled. We're talking about it not being coiled. So therefore, because it's not coiled, it's a straight chain. And that straight chain contains the 1 to 4 glycosidic bonds. That would therefore give us D as the right answer. So we're eliminating the wrong answers and leaving only those right answers. Okay, moving on to the next one then. Which nucleotide is shown in the diagram? Interesting. Right, straight away, I would be saying, right, there's my phosphate group. There's my pentose sugar. And there's my nitrogenous base. So I can see that this is a single ring. So because this is a single ring, I know this is a pyrimidine. And if it's a pyrimidine, that tells me it has to be either thymine or cytosine. If we are looking at DNA, if we're looking at RNA, then it would be uracil. So straight away, I need to note whether this pentose sugar is a deoxyribose or a ribose sugar. Now, I can see at carbon number 2, there is an oxygen present, which means it's not a deoxy. So I can remove, that's a 50-50 shot. I know it's an RNA molecule, and the RNA molecule, therefore, has to have a pyrimidine base. I've already said it's a single ring, and that would therefore leave me with the correct answer, which is T. Okay, question 16. Which statement is a correct description of the structure of ADP? So ADP is a adenosine diphosphate. So if I'm looking at this, I therefore know that I have a ribose sugar. I have an adenine. So this is the adenosine part where I've got the ribose sugar. Adenosine. Now with one phosphate, that would therefore be called AMP, adenosine monophosphate. If I add a second phosphate, I now have ADP, adenosine diphosphate. And if I add a third phosphate, I now have ATP, adenosine triphosphate. So that's, first of all, just me blurting down on paper what I know. I've got an adenine with a ribose sugar and whether or not I've got 1, 2, 3. Now, they've told me it's a di because they've told me it's ADP, adenosine diphosphate. I know that I've got adenine. Now, adenine, I know, is a purine, not a pyrimidine. So I can remove A being incorrect because I know that thymine and cytosine are pyrimidines. Therefore, adenine has to be a purine because an adenine and a thymine pair together, a purine and a pyrimidine always pair together. So therefore, I can remove that one. Now, I know that it's adenosine. So as I've already said, it's a ribose sugar. It's not a deoxyribose. So again, I can remove B. So it's really important that as you're going through this, that you're double checking what it is that they're saying. So this is quite a nice paper, I would say, for the AAS side of things. And then we can see, right, well, we know it's a diphosphate, which means it's got 2, so it can't have 1 phosphate group present, which therefore leaves us with that it's a nucleotide. It is made up of a nitrogenous base, a pentose sugar with a phosphate attached, but it has 2 phosphates. It's still a nucleotide. Okay, question 17 then. So a student investigates the effect of pH on the activity of the enzyme catalase. Oxygen is one of the products. So we've got catalase. Catalase, I know, is going to break down hydrogen peroxide into water and oxygen. So then because I know it's going to break it down into water and oxygen, I'm now looking at what else are they telling us. Student measured the volume of oxygen produced over a 10-minute period. Use this data to calculate the rate of oxygen production. Okay. Which row in the table shows the different types of variables? So independent variable is what they have changed. So a student investigates the effect of pH. So therefore, they're going to be changing the pH. So that would therefore eliminate C and A because I know that pH is what I'm changing because they told me I'm investigating the effect of pH. Now, my dependent variable is what I'm going to measure. So this is measured in a response to what I changed in my independent variable. So therefore, because that is now eliminated out, because that moves across, what that therefore means is I'm now down to the dependent variable being the catalase concentration or the volume of oxygen. Well, they already told me the student measures the volume of oxygen. So again, a lot of the time, the answers are there if you take your time and read the question properly. So because of that, that would leave us with the right answer, which is D. And let's just double check. What are they going to control? Well, they want to control the catalase concentration because they want to control everything else. So control variables would be everything else like temperature, like volume of hydrogen peroxide, concentration of hydrogen peroxide. So again, whatever they're asking, you need to make sure that you are aware of where they could go, because just because they've asked it this year like this doesn't mean they won't flip it and then ask it as a 2-3 marker, which is something they do tend to do quite a lot. Right, the student determines catalyst activity by calculating the rate. So we know, because we're looking at the rate, we're going to be looking at the volume over time. So that's important that we understand that it's volume over time. So therefore, we now need to look at, well, what's the appropriate units of measurement? Well, normally we measure our volume in centimeters cubed or decimeter cubed. So let's just have a look at what it says up here. We're doing it over 10 minutes. Are they going to measure decimeter cubed in 10 minutes? Probably not. So therefore, we can eliminate decimeter cubed as the volume they're going to collect. So they won't do it for long enough to collect that. So therefore, because we are eliminating the decimeter cubed, and also it should have been the other way around if we were looking at decimeter cubed, the volume has to be over time. So therefore, it has to be B, which is centimeters cubed per second. Okay, 19, then I'm going to finish at question 20. So what have we got? Which statement is true of stem cells? So this is module 2, 2.1.6, and this will come through quite a lot. So all stem cells can naturally divide to produce a new organism. No, not all stem cells. We're producing a new organism. They're only the totipotent stem cells. Stem cells are try that again. Stem cells are differentiated and specialized cells. No, they're not. They're undifferentiated. They have the capacity to go through mitotic division and differentiate. So we're eliminating some answers here because we know straight away that it's not those two. So we're now turning to a 50-50 shot. Stem cells are found only in early embryos. No, that's embryonic stem cells that are pluripotent, but we have multipotent adult stem cells. So therefore, that one is not correct. So let's just double check. Stem cells in adult humans can divide into, yes, they can, because they're multipotent. Why are you teaching in the summer vacation? So I am teaching for a little, excuse me, a little period, because there's still year 12s who need to do that revision. I will be taking a break in the summer months, but right now, whilst I'm still working, I decided while schools are still in the UK, students are still at school, I am offering to do the work because actually I want it to be that it helps students, and also these videos that I do right now are going to be saved on my YouTube profile. So therefore, people can watch them at any point throughout the summer if they want to do the revision, they want to catch up, or they want to just learn something new. This is why it's there. Just because it's summer vacation doesn't mean I have to stop or that I shouldn't do this. But at the same time, I will take a break. But at the moment, I enjoy what I do. I like doing this. Not for many students, to be fair, but it's not about those who are here right now. It's about who I can help when they come across my channel. Right, going to finish on this one though. Which of the options is an adaptation of a hydrophyte? So this is checking your understanding. What's a hydrophyte? Phyte means plant, hydro means water. So we have a plant that's found in a lot of water. So what can we be sure of? Well, why do plants need thick waxy cuticles? So the thick waxy cuticle is to prevent dehydration. Does a plant that lives in water need to prevent dehydration? No. So they don't have a thick waxy cuticle. They have a thin waxy cuticle. So it's not going to be A. Then we have fewer leaves, which result in a reduced surface area to volume ratio. Well, if they have fewer leaves, they're not going to be able to photosynthesize, and that would reduce their growth. So what you tend to find is that they have large leaves. And those large leaves are floating on the surface so that they can maximize the amount of light that they're absorbing. So it's not that they have fewer leaves, it's the fact that they are going to have large leaves, and that would not be for a hydrophyte, that would be a xerophyte, because the leaves are the place where they're going to lose water through transpiration. So the fewer leaves are going to be more for a xerophyte adaptation because they don't want to lose too much water. Okay, so this last one, many stomata on the upper surface of the leaves or storage of water in parenchyma tissue in the roots and the stem. So again, this is very much a, do you know the difference between your xerophyte and a hydrophyte? So a xerophyte is where there is no water or limited water, excuse me. So a plant that has to be adapted to where there's no water. So therefore the storage of water is not required in a hydrophyte because they live in water. So therefore leaving you with the only right answer, which was C. Okay, I'm going to finish there for tonight. I hope you have enjoyed my session. I will come back again next week and we'll look at the next set of questions. Thanks, guys.